Hello everyone. In this lecture video, I'll be talking about southern. The southern blotting. This was developed by the scientist Edwards M. Southern in 1975. The southern blotting is nothing but the hybridization technique which is used for identification of a particular size of DNA from the mixture of other similar molecules. This technique is mainly based on the principle of separation of DNA fragments by gel electrophoresis. And then these separated fragments are identified by using the label probe hybridization. Basically, DNA fragments are separated on the basis of size and charge during electrophoresis. Once these fragments are separated, these fragments get transferred into the nylon membrane and then those fragments are fixed over there. Then the DNA in which we are interested, it is detected by using the specific DNA probe, and these probes are obviously complementary to the desired DNA in which we are interested. The hybridization probe is nothing but the small single-stranded DNA, and the length is usually 100 to 500 base pair. And these probes are labeled with a marker so that those probes can be detected after they hybridize with the DNA in which we are interested. Let's see the process of blotting a southern blot. This process involves the first step would be the gel electrophoresis. You can see this is the well where these are different wells where we load the DNA sample. Then we run the agarose gel electrophoresis. After this, you can see this is the direction of migration of DNA towards the positive charge, because DNA has got the negative charge, and once they get migrated up to the opposite end, then we stop this process, and the next step would be a blotting technique, where whatever the DNA fragments which have formed the band on this gel, so this DNA get transfer onto the um, nitrocellulose filter. You can see this is the nitrocellulose filter, and this transfer is known as the blotting technique, where the this is the salt solution, and the salt solution, it passes through the gel and then through the filter up to the paper towel. These are nothing but the tissue paper towel. So by means of capillary action, this salt solution, it moves in the upward direction, and while moving this, it carry away the DNA fragments which are there on the gel, and because of that now it got transfer onto the nitrocellulose filter. See, you can see this is nothing but the nitrocellulose filter, and the DNA got transfer on this filter from the gel. Now the next step would be fixing of this transferred DNA. So that is carried out by baking in oven or by using different methods. And next is we need to identify the specific DNA in which we are interested, and for that we use the probes which are labeled, and these probes are single-stranded DNA. These probes are allowed to hybridize with the DNA which are there on the nitrocellulose paper, and then whatever the probes which are not involved in hybridization, I mean the probes which are unbound, those can be removed by washing. And the next step will be the identification of those probes, and ultimately the DNA with which these probes have formed the a complementary relationship. Being a complementary in nature, they hybridize with the DNA, and that can be detected by using the autoradiogram. So this is the way by which we can identify the DNA in which we are interested. Here the main thing is that the probe and the target DNA are complementary to each other. The reaction is very specific, which helps in detection of the specific DNA fragments. Let's see each step in detail. First step will be extraction and purification of DNA from the cell. Here the DNA is first separated from the target cell following the standard method of genomic DNA extraction and then purification. Then the restriction endonucleases, these enzymes are used to cut the high molecular weight DNA strands into the smaller fragments. So this restriction endonuclease enzymes helps us to cut the DNA into smaller fragments. Then one or more restriction endonuclease can be used in order to get these kind of fragments. Then whatever the fragments which we get after the restriction ligations, those fragments are separated by agarogel electrophoresis, where being a negative charge molecule, as the DNA has got the negative charge because of presence of phosphate group, it moves towards the positively charged anode, and the distance run by the DNA, that all depends on the charge and the size of DNA. Next, the DNA which are obtained are double-stranded in nature. Now we need to convert it into single strand. Otherwise, how it will be able to form the hydrogen bond with the probe, isn't it? And therefore, alkali are used to denature those restriction fragments which are there in the gel. And because of alkali treatments, what happened, whatever the double stranded DNA which are there in the gel, those double stranded DNA get converted into the single stranded one because of breaking of hydrogen bond. Now again, there is a possibility that this single stranded DNA may rehybridize, means they may again form the hydrogen bond with each other, being a complementary in nature, and therefore we use NaCl so that the DNA get neutralized, and this will help us to avoid the rehybridization. Next step would be the depurination. Actually, this step is optional. See, if you are handling the fragments which are smaller than 15 kilo base pair, then this step is not necessary. But if the fragments are greater than 15 kilo base pair, then it is quite difficult to get transferred to the blotting membrane, and therefore depurination is necessary. So depurination with HCl, it is carried out, and which simply takes the purine out, and because of that the DNA fragments get converted into the smaller fragments, so it becomes easy to get transferred to the blotting membrane. Next step will be blotting. Here the denatured fragments are then transferred onto the nitrocellulose filter paper, and this is done by placing the gel on the top of the buffer saturated filter paper and then laying the nitrocellulose filter paper on the top of the gel. You can see this diagram. This is the container, and where it contains the buffer, okay, and in that tray we have kept the platform, and then Whatman paper is placed in that buffer, and by means of capillary action, whatever the buffer which is there in the tray, that will move in upward direction. Next to this Whatman paper, there is a gel which is shown purple in color, and immediately above the gel there is a membrane, that is either the nylon or nitrocellulose membrane is placed. Above that membrane, there is a Whatman paper stack followed by a paper towel stack and then the weight. Now, because of capillary action, what happened? Whatever the buffer which is there in that tray, that will move in an upward direction. Okay, and while moving in upward direction, what happened? It also carries the DNA fragments which are there on the gel, and thus the DNA fragments get carried away by the buffer, and it get transferred to the membrane. Okay, next is this process is known as the blotting, and mainly it based on the capillary action. Next, now the nitrocellulose membrane is removed from the blotting stack because now it is with the DNA strand, isn't it? Now the membrane with single-stranded DNA bands attached on it, we need to fix those DNA bands on the nitrocellulose paper. Okay, and therefore it is baked in a vacuum, or it is kept in oven at 80 degrees Celsius for 2 to 3 hours. And because of this treatment, those bands get fixed to the that nitrocellulose membrane. Now, another technique is we can expose it to ultraviolet radiations so that it helps for the permanent attachment of that DNA to the membrane. Next step will be the hybridization. Here, the membrane, which is then exposed, the membrane on which the single-stranded DNA is fixed, that is exposed to the hybridization probes. And these hybridization probes are nothing but the single-stranded DNA fragment with a specific sequence, and those sequence would be complementary to the target DNA, which we need to determine, which we need to identify. Okay, now the probe DNA is labeled so that it can be detected, usually by incorporating the radioactivity or tagging the molecule with a fluorescent or chromogenic dye. So the probes are labeled by either of this way. Number one is we can incorporate the radioactivity, or we can tag the molecule with the fluorescent or chromogenic dye. And important is that these probes are complementary to the DNA in which we are interested. Next, after hybridization, the membrane is thoroughly washed with buffer. Why this washing is necessary? This helps to remove the probes that is bound non-specifically, or there may be presence of some probes which are not involved in bonding with the specific DNA. So those unbound probes can be removed by means of washing step. Now, last step is autoradiograph. Here, the hybridized region are detected autoradiographically by placing the nitrocellulose membrane in contact with the photographic film, which shows the hybridized DNA molecule. The pattern of hybridization is visualized on X-ray film by autoradiography in case of radioactive or fluorescent probe is used, or by the development of color on the membrane if chromogenic detection method is used. Means once the hybridization is done, now we can detect the specific DNA in which we are interested with the help of probes which are radioactively labeled. If we have used the radioactively labeled probes, in that case we can go for the autoradiography. Okay, or if we have used the chromogenic I mean if we have used the some color to that probes, then we can go for the chromogenic detection method. So this is the way by which we can find out the DNA of specific sequence. We can find out the DNA in which we are interested by using the southern blotting technique. Now, what are the applications of the southern blot techniques here? As I said, it helps to identify the specific DNA from the DNA sample. Suppose we are having the 100 different fragments, then from this 100 small DNA fragments, we can identify the one in which we are interested, having the specific sequence. Next, identification of viral and the bacterial infection. Yes, so we can go for the specific gene, presence of specific gene for the identification of particular viral and the bacterial infection. It is also used in the forensic study, which helps for the identification of individual. Then it helps in the study of gene mutation, as well as the southern blot technique plays important role in the diagnosis of neonatal and genetic disease including cancer, because in this case you will find the change in the nucleotide sequence, and this altered nucleotide sequence can be identified by using the southern blot technique. Thank you.